starts now, and I can see that there are some questions. Oh, thank you, Balkis, for letting me know because I was not recording. Okay, so as I said, the discussion about the transgenic animals will be on two different sessions. The first session is going to be discussing the basic transgenic animal and the general knockout mice. In the next session, then we will move into generation of a knock-in mouse and inducible tissue-specific animals. And I was saying that in the steps of generation of a transgenic animal, we need always to start from the basics and those techniques that we learned, which are identification of a gene and then cloning a gene into a plasmid under a specific promoter. And as you will see later on, it's going to be the first step of a transgenic animal generation. Now, in the previous lecture, I gave you one example, which was, imagine you want to overexpress a gene, and we took that gene as an example, insulin. So we needed to have a promoter. In this situation, it's a cytomegalovirus promoter. And we insert the cDNA for the insulin downstream of this promoter. And then we provide with a poly-A signal to get a stabilization of the RNA. Okay? And then we discussed one of the examples, which was the expression of green fluorescent protein in a mouse under the control of ubiquitin C promoter. And as the name indicates, ubiquitin is a protein that is expressed in all the tissues all the time. And therefore, if you look at the mouse tissues at any level, from the embryonic development all the way to the adult, you can see the green fluorescent protein is being expressed in all the tissues. So before moving into the transgenic animal generation, I would like simply to review with you the models that we can use in the biomedical research. And you will see that this course is going to give you a background almost on 3 or 4 of these models. And 2 of these models we are not really touching very deeply. The first part of these models is that if you want to study a gene or a disease, the first thing you need is an in vitro model, which is the primary cells or the cell lines or the organoids. So you will do a lot of in vitro studies, and these are the in vitro models that we use. And then probably you can do some ex vivo models, and these are probably tissues that you obtain from diseased animals or from diseased patients. So you can grow those tissues and you can perform experiments. And today we will move from these in vitro models that we discussed in cell culture and transfections and in overexpression of certain DNA in cells into the animal models. In between these two, after generating transgenic animals, there is one model that we call humanized animal models, and these are mostly mice that contain a gene or express a gene that comes from a human. So say, for example, you want to know what's the role of a novel gene that you never studied. You take that gene, you overexpress it in mouse, and you would study how that gene behave in mouse. Of course, there is a lot of in silico models that comes from algorithms and bioinformatics and AI data that we could run analysis and guess what is the gene gonna perform. And all these 5 models are essential to go to clinical trials. So we will do tons of research in vitro on animals and bioinformatically, and then only very few candidates that will move into the next step into the clinical trials. Okay, so if we look at the in vitro models so far, the primary cells, you know that these come directly from tissues, like from heart, from liver. You can simply take the tissue, dissociate it, and put it in culture, and you use it. But these primary cells, they can't last forever. That's why we have the cell lines, because these are immortalized. And we discussed in the assignment and in the first two lectures, the HeLa and the HEC293 cells. They can be cultured indefinitely. And we can use them to do subcellular localizations, cellular processes, drug development, screening, and gene expression. We could go in the cell culture into another level by looking at 3D cultures, where we put the cells, instead of only in a culture dish, we can put them to make some sort of organs in 3 dimension, and that would reshape somehow the architecture of a tissue. Now, in terms of the in vitro model, there are cells that are really special. These are the stem cells. The stem cells, because they are very important in terms of their potential for differentiation, because pluripotent stem cells can differentiate in principle to any cell type. Their culture is different and their usage is different. So we will need to know how to culture embryonic stem cell in order to be able to generate a transgenic animal. So the stem cell comes like a transition between the in vitro models and the transgenic animal generation. As you know, nowadays we have induced pluripotent stem cells, and these are very widely used in research. Almost any single publication that comes out now, it has some sort of relation with the embryonic induced pluripotent stem cells, particularly those which are working on regenerative capacity of brain, regenerative capacity of heart, and drugs for those particular tissues. Now, the ex vivo, as I said, it's a tissue explant. In other words, you would take the tissue slices and you would retain some components or properties of a tissue, but at the same time study their functional properties and study how they react in terms of responses to an injury or a treatment. Now, the third level is going to be the in vivo models, and that would be many types of models, not only mice. In our lectures today and tomorrow, we are focusing on mice because it's very widely used. But that being said, there are many other models that can be used. Mice is one of them, rats, rabbits, zebrafish, pigs, and dogs. So at least these are the animals that we use in the lab. Why mice are the most widely used? Because first of all, they are smaller in size. You can cage them. So these are individual cages here. It can take up to 5 animals, depending on the size, 4 animals maybe. The mice life cycle is very short, if I want to say, in terms of the fertilization and embryogenesis. It takes around 21 days from the fertilization process to the birth. So that's relatively very short, and it's very good to perform experiments. You don't want to wait 3 years to perform an experiment. And within 5 to 6 weeks, they reach maturity, so you can perform experiments on those. That being said, they have a homology around 88% with the coding DNA with the human, so around 12% differences. And exception for this is that the non-coding part of the mouse is way different from the human. So all these characters makes the mouse one of the most useful or the easiest model to utilize. And you can know, like, if you go to any institution right now, you will see an animal facility where you have mouse and rat are being extensively studied. So if we would like to generate a transgenic animal, what do we need to do? As for today, by the end of the day, I hope you will be able to process all the information that is needed for generating a transgenic animal. Now, with little training at the bench, you will be able to eventually amplify a gene and prepare it to generate a transgenic animal. Obviously, some of these steps are very specialized. You can't do yourself. You can send it to a core somewhere, particularly, for example, the injection of DNA to the nuclei and the embryonic stem cell manipulation. So transgenesis, as I mentioned before, is defined by adding a foreign genetic DNA into an organism's genome. So this means when you say a foreign genetic DNA, this means probably a piece of DNA that comes from one mouse that you inserted in another mouse, or from human into a mouse genome, or a virus to a human genome, or into a mouse genome, sorry. And you have to retain this gene for many, many generations. So across every generation that comes from that mouse, it will be, the transgene will be transmitted. Of course, we are trying to understand the role of certain of these genes. So introducing this gene will introduce a new function that is performed by this gene, and that mouse model will be used to study that foreign gene. And in general, transgenesis means gain of function. So many times when you overexpress a gene, you will gain the function of that gene. So in terms of transgenesis, it's widely accepted that transgenesis means gain of function. Not 100% exclusive, but widely accepted. So an organism that has been permanently altered by adding a DNA sequence to its genome, this is a transgenic animal. And the foreign DNA that has been carried or transmitted, that's the transgene. Now, I mentioned before that humanized animal models, mostly these are mice that are genetically engineered to express human genes or carry human cells. For example, a tumor that we find in human and we want to study, so we simply take the cell line from that humor or cancer cell line and we can insert it in mice and we can see how the tumor behave. This is also a humanized model. So therefore, these models are very useful to understand gene function and responses in general for immune responses, for cancer responses, infectious diseases, and certain drugs in terms of how they change the metabolism or stuff like that. Okay. Now, in terms of a knockout animal, it's a transgenic animal, but we are disrupting an existing function by blocking a specific gene. In other words, in that animal, there is a gene that is built in the genome. However, we are knocking out that gene, so we will lose the function of that gene. And genetic knockouts are very important to understand the physiology behind certain genes. So we would like to understand what, if in conditions that we lose that gene, what would be the physiological response. Obviously, we can do this in a human, so animal models are the perfect model to test. Okay, one of the examples I'm showing you here is the leptin knockout mouse. And for those who don't know what is leptin, this is a hormone that helps to maintain the body weight. It does multiple functions in different tissues, but the most important of those is that the levels of leptin in your body, in your blood, is directly related to the body fat content. In other words, if you lose leptin, the animal, or in general, if there is any compromised function of leptin, the body fat content increases. So as you can see in this image here, this is a leptin knockout mouse, and that's the wild type litter. So these are like the same litter, so they're brother and sister. This is a wild type and this is a knockout. And you can see that the accumulation of the fat is due to the loss of leptin. So obviously this mouse is perfect model to study the effects of leptin on the body fat content and the induction of, or the increase in the body weight. Recall from the previous lecture that we discussed the mitosis and the meiosis. And I mentioned that when we are gonna generate a transgenic animal, we have to understand the process of mitosis and meiosis and how we can utilize the end product of mitosis and meiosis in terms of the transgenesis. So recall that mitosis happen in somatic cells, and the end product is that one cell is dividing into two identical cells with the same amount of genetic material. While meiosis happen in germ cells or gametes or sex cells, and it will generate at least n equal 4 in sperm, it's n equal 4, but in eggs, it's only 1 due to other factors. But the process of meiosis is the same. Bear in mind that meiosis in males and females is totally different. While meiosis takes place within the same period of time in males to generate the sperm, and you get 4 sperm of only half amount of the DNA. So recall the parent cell has 2 N chromosomes, and the resulting sperm in condition for a male, you will have 4 cells each of only half the amount of the genetic material, which is only 1 N, not 2 N. Now, in females, the process of meiosis starts before birth and ends in puberty. Be careful that this is totally different, and that's why there are other factors to be concerned, to be considered, and only during the maturation of the egg, only one of the eggs will be matured in order to be prepared for fertilization. So keep this in mind. Now, the end product of meiosis is n equal 1. So meiosis generate one N genetic material in a gamete, and the fertilization comes where the egg and the sperm would meet in order to reconstitute back the 2N component of the genetic material. And therefore, this is how the life continue. You generate 1N, and therefore you are in need to recompensate for the another 50% of the genetic material, which is the 1N. You can get that by the fertilization process. So fertilization restores the full chromosome number by combining the genetic material from 2 gametes to form a zygote. Okay. Obviously, some of these things that I'm mentioning to you should look like very trivial, but I will tell you why I'm coming back to this in a second. Now, immediately after the male, immediately after the penetration of a sperm into a fertilized egg mouse, there should be 2 pronuclei. One pronucleus come from the sperm, and one that come from the egg. And therefore, these 2 pronuclei at certain point have to fuse to form the zygote nucleus, which will eventually grow to become the embryo. So this is important because timing is very essential to understand where we can interfere in order to generate a transgenic animal. And this will come in the next couple of slides. Now, one thing you have to keep in mind that the male pronucleus is typically larger than the female nucleus. Therefore, if we would take an egg that was just fertilized few seconds or few minutes ago we will directly know that the pronucleus that comes from the male is a little bit more different from that one of the female. And I'm not sure you can see it very well here. Obviously, scientists who are experienced in the lab, who are doing this experiment daily, would know that this is the pronucleus from the male, because it's less condensed. You have a bigger size compared to the pronucleus of the female. So why this is important? Because in the process of generating transgenic animals, we have the 2 or 3 different methods that we can use. And each of these methods is depending on when you are going to start the transgenesis. In other words, if we want to express a gene that is random, that is inserted anywhere in the genome, then we would take an oocyte that is just fertilized by a sperm, and then we will insert the DNA directly to the pronuclei of the sperm. And then we will let that egg grow from the 2-cell to 4-cell stage to 8-cell stage and get implanted in the uterus and get the transgenic animal. In another approach, in fact, we will use stem cells that come from the trophoblast or the blastocyst, which is one of the stages after forming of a morula. So the stages would be an egg that is fertilized, and now you have 1 cell with 2 pronuclei, and then they fuse, and then after that divide into 2 cells, 4 cells, 8 cells, 16 cells, and that's probably the morula. After that, it will be a blastocyst, where you have some sort of cavity, and there is embryonic stem cells around that. When we perform the knockout animals, we use this stage of the embryo. So we have to take the embryonic stem cells from here, manipulate them, and then bring them back. When we generate a transgenic animal, we will get an egg that is fertilized, and we will inject the DNA in the pronuclei. So these 2 embryonic development stages are being used in 2 different modes of transgenesis in down the road. So probably I will discuss only 2 out of the 3 approaches. So what are the methods? Somebody asking. Gary, you are asking, is the knockout still considered if no foreign genetic material is introduced? Well, as I said, transgenesis is mostly and widely accepted to be an overexpression or an introduction of a gene. Okay. But the knockout, because you are kind of modifying the genome, you are not introducing something new but you are still modifying the genome. So it's still a transgenesis, but it's a specific type of the transgenesis. So we will discuss 2 methods, the DNA injection and the embryonic stem cell approach. So in the DNA injection, as I said, we will get a fertilized egg where it's like just the fertilization took place within like less than 24 hours, I would say. And then we will inject the transgene into the pronucleus, which we know that is bigger from the male. And why I'm saying it's injected to the male, one of the logic behind that, because the pronuclei from the egg will fuse with a foreign DNA. So therefore, there will be less issues if you inject the DNA into that foreign pronuclei. And after that, you will simply let this zygote grow. So eventually the fusion will take place. But now you have a foreign DNA that is introduced. And then you will generate embryos in an animal. So what you have to do after this process is that you will take those eggs and you plant them into a mouse where they can grow. And eventually you get a transgenic animal. So this is the DNA injection approach, and that's the most widely used in the transgenesis. Now, in case you want to knock out an animal, you would use the embryonic stem cell approach. And in this condition, you will take embryonic stem cells and you will transfect those with a plasmid that contains information about the gene you want to knock out. And I will describe this in the next couple of slides, how you do that. Once you transfected the cells on that plasmid, you should have some sort of selection. So you can select which stem cells being transfected with the plasmid and which one is not. And you grow these embryonic stem cells. And after that, you would take these and you insert them into a mouse embryo. So obviously, because you are at the step of stem cells, so this means you are already at the stage of more than 32 cells in the embryo. Because remember, the fusion here, it generates one zygote, one egg, and then it divides into two, and the two into 4 and 6 and 8 and 16, and the morula, and then the blastocyst. So obviously, you are introducing these back into an embryo in the blastocyst because you need that space in order to insert these embryonic stem cell. Those stem cell will get recruited to the embryo, and therefore, not all the embryo will contain the embryonic stem cells. And we will discuss this why. Once you get embryos from that mouse, so you would implant these embryos into a mouse, and this mouse will generate offspring. Some of the tissues would have stem cells that you have modified, and some would be not modified. That's why you have different color in the fur of the mouse. So I will describe these steps many times back and forth, and by the end of this lecture, you will be able to differentiate very nicely between a transgenic animal and a knockout animal. Okay, now before moving into the steps, so if we discuss the transgenesis, remember that if you want to overexpress a gene in culture, you need the promoter, and you need the gene, and you need the poly A tail. So you need the cDNA, the coding sequence of the gene, and then the poly A tail. Now, if you will introduce that gene into the genome, the first step you need is to linearize it. So you have to cut the gene from the beginning of that promoter till the end of the poly A tail. So you have to cut that piece. It's linear now, it's not in a plasmid. And therefore, you need to have, that's why I said the cloning process is the most important process in any of these steps, because you would need to know which plasmid you are using. You would need to know which restriction enzymes you have here so that you don't cut within your gene. Exactly like I told you in the assignment number 2, the last question, which restriction enzymes you would use. Definitely you will have the sequence from this to there, and your restriction enzymes should not cut anywhere here. It should be cutting only here and there. And now you will have your gene linearized. You will purify it, and then you can inject it. Okay? So this is one of the steps we need in the transgenesis in the injection procedure. So how the procedure takes? What are the steps? So the oocyte of the mice and other mammals, of course, these are large enough that we can inject DNA directly into those. So this is something that changed the history of transgenic animals, because if the oocytes were very sensitive for the injection process, we would not be able to generate transgenic animals this way. Transgenesis, as I said, starts with the cloning of the gene of interest, whatever gene is that. So you have to learn about it, study it in vitro, know the sequences, know the cDNA, know if there's multiple transcript variants, which transcript variant you would like to express. So all that has to be done in vitro way before you decide to do a transgenesis. And then you would need fertilized eggs that are isolated from mating of wild type animals. Meaning I would put male and female mice and they will mate and they will produce a zygote where males and the sperm and the egg just fuse, and then we will kind of steal it from that animal. So we will isolate it. And then the cloned foreign DNA, few hundred copies. So we will calculate how many copies we could know from the amount of DNA we will inject, will be injected into the sperm pronuclei just after the fertilization of the mouse and before the sperm and the egg nuclei fuse. Because if the sperm and the egg nuclei fuse, the efficiency will be much, much lower, and probably it will be lethal sometimes to inject DNA there. Recall that the DNA should be in linear copies, not in a circular copy. So we have to make sure that we cut our transgene and purify it before we inject it. Now, the injection of the DNA at this stage allows the foreign to get recruited into the embryonic cells, which is at this stage is a fertilized egg, and it will be eventually the future embryo. In other words, the embryo, any embryo that is generated, if you get one animal, two animals, five animals, like if you get 5 mice, 6 mice, probably there's a chance that all of them could be transgenic, or 30% transgenic, or 50% transgenic. In principle, whenever the injected DNA get into the chromosome and gets recruited, you will get a transgenic animal. And normally, the transgene are inserted in forms of strings of tandemly repeated genes. Recall when we discussed the tandem repeats, where it's a repeated sequence. In other words, you have multiple copies of that gene, one after the other. Sometimes 3 copies, sometimes 4 copies, sometimes 10 copies that are inserted one after the other. Now, the only disadvantage of this process is that few DNA copies integrate randomly. So you don't know. Obviously, we can know where they are inserted, but we cannot decide where they will be integrated in the genome. So they will be integrated into the chromosomes through non-homologous recombination, so it's unspecific integration site. So therefore, one of the issues that you can imagine is that probably our gene will be inserted somewhere that is essential and could create a problem to other genes. Okay, so this has to be taken into consideration. After the injection, the embryos, which are multiple fertilized eggs, are implanted into pseudopregnant female. So we take these embryos and we implant them into pseudopregnant females, meaning this is a female that we provide with hormones to be prepared in order to get pregnant. It's similar to the in vitro fertilization. We will discuss this in the last slide. So a pseudopregnant female is a surrogate mother that has been physiologically prepared by having hormones ready for the pregnancy. Okay? So if we look at the steps, now after reading the slide, you can probably imagine things much easier. So we have male and female mice. They mate, they will generate a fertilized egg. In that fertilized egg, we have a pronuclei from the male and one from the female. So the one that comes from the sperm, it should be a little bit bigger. It's not really emphasized in this image, but we discussed it. What we will do is that we will linearize our gene. We will know how many copies we will add. and then we will inject that gene into the pronuclei of the male or the sperm. And this will lead to the insertion of that gene into one of the chromosomes that we know it's a random, we don't know where. Then we let this grow, and then of course, we are not doing only one egg and one sperm. And when we do this process, keep in mind that we will get many eggs and many sperm. So we will get many of these fertilized eggs, because normally a mouse, a male with a female, when they mate, they will generate 6 to 12 animals. So we have a chance to have from each male and female mating 6 to 12 fertilized eggs. So if we are doing this experiment in the lab and we want it to work, we will mate 5 males and 5 females, and then we will have around 60 eggs that are fertilized. And we will do 60 injections of the transgene into those pronuclei, because you never know how many of these will survive. And then you will take around 30 to 40, and you will implant those into the pseudo-pregnant mouse. And then you will get, for example, this animal got 6 litters. So we will see how we will be able to say, oh, this is a transgenic animal and this is not. Okay. Now, out of these 6 animals, they are called A, B, A to F. Some of them are transgenic. In other words, some of them, they have recruited the DNA into their genome, and some of them not, because the recruitment didn't take place. So the transgenic animals have genomes that have been permanently altered through the recombinant DNA technology. So this is important, because if, for example, A has the transgene, as you can see in this blot, if it has this transgene, it has to be able to propagate it to any generation that comes. So whenever we mate this pup, it becomes adult, and we mate it, it should be able to transmit the gene to the next generation. In the applications that we discussed, like now, as for today, you will start finding some sort of logic of this course, because you will start using the techniques that we were just discussing as a technique in order for an application. So you will see the application here. So we discussed Southern blotting and we discussed PCR. So we can generate a probe that specifically picks that transgene, and we can use it in a Southern blotting, so we can identify which animal is transgenic and which one is not. Or we can generate PCR primers that specifically picks that transgene, and we can do a PCR so we take DNA from all these animals, and we do a PCR, and whenever we get a PCR product, then we know that this animal has the transgene and this one doesn't, because it doesn't have the transgene. And this is generally called genotyping. I don't know if some of you is doing anything like this in the lab. Now, the negative PCR results, okay, so sorry, I missed here. So we design primers to identify or amplify the transgene. In other words, whenever we do a PCR, we take the DNA, we put the primers, we amplify that product, and then we can see if the transgene is there or not. The transgenic animals are clear. This is A, C, and D, because they have the transgene in their genome. And if you take any cells from this animal, you will be able to do the PCR, the DNA extraction and the PCR. Now, negative PCR product means that the animal is not transgenic, and this is obvious, B, E, and F. DNA injection to the pronuclei is widely used for expressing transgenes. So this is one of the most wide, if you go to NCBI and PubMed and look for overexpressing genes using different promoters in animals, you will find thousands and thousands of papers. Okay, so if we put everything we have learned into practice, so I will just quickly recall how this course helps you to start looking for applications. So look at this particular experiment where we are generating transgenic animals. So what do we need to do? We need to identify the gene that we want to study. So we can go and get all this information from NCBI, Ensemble, the transcripts, the variants, the coding sequence, the upstream, the downstream of the gene. You need to clone that gene into a vector. So now you know how to clone that gene to a vector in a plasmid. And maybe you can use the PCR approach to do that simply by taking the coding sequence and perform a PCR where you amplify the coding sequence as we did in the assignment 2 in question number 9. And then you can, after cloning that gene into the plasmid, transform the bacteria so that you can amplify that plasmid. And the amplification process by growing the bacteria and getting copies of that plasmid. And after that, you can isolate the DNA, as we discussed by the different ways of isolation and the maxi-prep. And then you can digest by the restriction enzymes the transgene and the promoter in order to linearize it. And you can use the PCR strategy to identify transgenic animal from non-transgenic animal. So everything we discussed in terms of the DNA, you can just say like you did it all in this experiment here to validate, to generate transgenic animal and validate the results you get. Okay, so I want to stop here for a couple of minutes if you have any questions. Otherwise, I will continue. Any questions? No questions. Great. It was very clear then. I will take it as it was very clear. So the second type of transgenic animal or introduction of a gene, transgene, it would be a knockout process. The knockout process, and one of your colleagues asked if knocking out a gene is still a transgenesis. Yes, it is a transgenesis in a sense that in order to knock out that gene, you have to insert something in its place. And I will be describing this soon. So in this approach, recall that I want to go back a little bit on the slides. So recall that the DNA injection, it happens at this stage, just after the egg has been fertilized by the sperm, within the first 24 hours of the fertilization. The knockout is going to happen at this stage, okay, from the blastocyst and up. So what are the advantages of performing this type of transgene introduction? So the approach of using the embryonic stem cells is mainly dependent on the fact that the embryonic stem cells can differentiate to any cell type in the mouse. And therefore, we can use the second part of the advantage, which is we want to specifically knock out a specific gene. So we have a specific gene that we want to target. And obviously, this is the biggest advantage over simply just injecting DNA into the genome that you don't know where it's going to be inserted and how many copies it will be inserted. You can't, as I said, we have techniques to know where it's inserted, but we can't really decide how many copies and where it will be inserted. And this procedure is divided into two steps. The first step is to create embryonic stem cells that has an interruption in that specific gene. And then we have to replace that interrupted gene in the animal with the wild type gene. And after that, we have to mate the first, second generation to generate that homozygote knockout. Now, probably some of you have not taken genetics to explain what is the homozygous recombination. I will discuss this in the next slide. So the first step in this procedure is first, we mate male and female animals. These are wild types. We didn't change anything in those. The female becomes pregnant and it has an embryo. It becomes at the blastocyst level. We will isolate the embryonic stem cells from this blastocyst. We take them into cells and we put them in culture. And at this stage, we will already have generated our transgene in the vector. So at this stage, again, we will need the vector to be already studied. We know what we want to do. We know the sequences, everything in that vector, we know it. We will simply transfect the embryonic stem cells into the DNA into the embryonic stem cells. And now, because that vector gives me some selectivity, I can select which embryonic stem cells are transfected and which one is not. The second stage is that, again, I will mate male and female animals. They will have a wild type blastocyst. And when they reach that point, I will introduce back the modified embryonic stem cells. And after that, we will leave the cells to grow, and we will go in the steps of generating the first and the second generation of those animals. Now, in the process of generating two different embryonic cells from two different animals, so this you can see it's kind of grayish mouse, and this is a white mouse. So the embryonic stem cells from this mouse is different from that mouse. And also those cells carry a transgene that is in red. So when we add these cells here, we will have a mix of two embryonic stem cells. And the production of a chimeric embryo or chimera is by aggregation of two different embryonic stem cells. One that comes from this animal and one that comes from that animal. Okay, so let's first discuss the the homologous recombination. And homologous recombination takes place in our body as a process to generate a more variability in our genetic material. In other words, at the time of the meiosis, the homologous recombination is mostly known as the crossing over, where similar DNA fragments recognize the homology. For example, this area here is homologous to that one. So they will know each other. And during the process of the meiosis and the segregation of the genes, some part of this green chromosome is flipped into that violet one. And the recombination process ends up generating these variable parts where one chromosome has a part from the other chromosome. So if we take this as a paternal and maternal gene, we have one chromosome from the paternal origin and the other from the maternal origin. And during the meiosis process, this can generate more variability where we can get genes from the chromosome comes from our mom and our dad that flip-flop parts of these into each other. Okay? I'm not going to ask you how the recombination process takes place. This is a pure genetics, but I have to explain that to you in terms of the course. Now, if we look closer how the process of homologous recombination take place, this is an example for you here. So this is the genomic DNA. So this is the chromosome, and this is the vector which we are going to use to introduce a specific gene, or a specific exon, or a specific part of a gene, whatever. You would need to have homology arms. So this sequence is exactly identical to this, and this sequence is exactly identical to that, so that the process of crossing over can take place. And what I would do is to engineer that vector to introduce whatever I want in the green that should replace the blue. So the end product would be that we replace the blue part of the gene with the green part of the gene. Okay, so that's exactly what we're gonna do, but I will go in more details in the next few slides. So this is a practical example from a paper where they wanted to knock out a specific gene. Recall that if, for example, you have this gene start with exon, so this is the wild type gene in the genome. It has exon 1, exon 2, exon 3, and exon 4. Recall that we said the cDNA can make exon 1, 2, 3, and 4, so this is one isoform. At certain cases, and most of the time this happens, if you remove one of the genomic DNA, the gene will be destabilized. So it will not be any more stable, and therefore it will be deleted. So we take advantage of that. And in order to delete, for example, this gene, what we have to do is to generate a vector that has exact same sequence of exon 1, exact same sequence of exon 2, but it doesn't have exon 4 and exon 3, sorry, and it doesn't have the exon 2 and the exon 4. Okay, now the homologous recombination, since exon 1 exists on the vector and on the chromosome, and exon 3 as well, so this would be a perfect site for the recombination. And if the recombination takes place, then exon 2 will be replaced by a neocassette, which is an antibiotic resistant gene. And then you will end up what? Having exon 1, and then you have an interruption with this antibiotic cassette, and then exon 3, and this will destabilize the gene, and therefore you will lose the gene function. So this is the basic concept, how we generate a knockout animal. Is simply by removing part of a gene. It should be essential. And anyway, as I said before, you should already have done a lot of in vitro studies to know that exon 2 is essential, and if you remove it, you're gonna get rid of the gene. Otherwise, you don't do it directly in the animals. Maybe you have to remove exon 3, or maybe you have to remove exon 2 and 3. So to decide which exon, you don't really directly go blind. You have to do a lot of in vitro studies. But once you know, then this is the strategy. You have to have the homology arms, which are represented here as exon 1 and 3. And the exon you want to replace is number 2, and you replace it by a neocassette. And this way, you end up having a destabilized gene where there's a separation between exon 1 and exon 3, and therefore the gene is going to lose its function. Okay, there are some challenges. Obviously, sometimes you remove exon 2, but still the gene can be functional. Or sometimes there might be a transcription for the exon 3 and 4, so you get a side product of that gene. Again, all that has to be studied in vitro before you go into the transgenic animal. And you have also to consider those challenges after having a knockout animal. Even if you achieved removing the exon 2 and have the neocassette, you still have to consider probably that there are some truncated version of the protein. So you have to test this in your transgenic animals. Okay, so now we go back to the strategy. It would be first creating the embryonic stem cells that come from the wild type animals. And then we have to introduce that vector. So that vector is called, the replacement vector. Okay. So that vector that we will use is called the replacement vector. So again, we have the homology arm 2, and then we have the gene where we will be replacing. And once we know which gene we are gonna use, which plasmid we're gonna use, then we engineer this part homologous to the endogenous chromosome. We will again cut it in linear and perform the process of homologous recombination. Okay, now let me just go back one step here to repeat the two particular steps. First, taking the embryonic stem cells from wild type, and then we transfect our plasmid into those embryonic stem cells. After that, I'll come back to describe this step here, which is step number 2. Now, what are the selective markers that I will use to make sure that the homologous recombination worked? So first, as I said, you will clone the gene of interest into the vector. Or I give you an example here. For example, the HER2, we know that it causes cancer, and we want to know, for example, if we remove it from a mouse, would we have less cancer or less tumor generation in that mouse? We have to interrupt the gene by using the neocassette, which is conferring resistance to antibiotic neomycin. And then we will introduce a marker, another selective gene. This is the most widely used. It's called thymidine kinase, just to know if the recombination is specific or not. And you will see this in the next slide. And after that, we will take these engineered embryonic stem cells and we will introduce them back into a pseudopregnant mouse. So let's go to the steps one by one. This step starts after mating male and female animals. So this is a wild type animal, this is a wild type animal. We made them together. They generate a zygote, and the zygotes start to grow, and they have embryonic stem cell. At this point, we took the female, and we isolated the embryonic stem cells from it. Okay? At the same time, we already have our gene of interest that we want to study. We know it here in green color. And at the extremity of that gene, we put the thymidine kinase. So it's away from the gene. It's like downstream of that gene. Then we interrupted the gene by introducing the neomycin cassette in the site. If you want to make it easy on you, let's imagine it's exon number 2. We removed exon number 2 and we introduced there the neocassette. Okay, now I have a plasmid where the transgene, the gene, is interrupted with there's no exon 2, there's only the neocassette. So it can have a neomycin resistant, and at the same time, it is sensitive to a drug that is processed by the thymidine kinase. Great. Now I will electroporate these vectors into the embryonic stem cells. So some embryonic stem cell, what will happen now? When the DNA is going to be integrated in the embryonic stem cells, there are 3 different outcomes. The first outcome would be there is no recombination at all. Therefore, the genomic DNA doesn't retain anything from the vector. Okay? In this situation, if we add neomycin, the cells will die because it doesn't have any resistance. So no recombination, no insertion of the vector or the gene into the genomic DNA, and therefore there is no resistance being conferred by the vector, and therefore the cells will die if you add neomycin. Okay, if the recombination take place, there are two possibilities. The first possibility is that the recombination is not specific. In other words, well, let me first explain the specific recombination because it's easier than to understand that one. So if the recombination is specific, this means on the genome side, the gene in the green we want to replace, this homology arm and this homology arm recognized perfectly on the genome and on the plasmid. And then if the recombination took place, there will be change. The genomic DNA, which is here, for example, exon 2, is going to be taken back to the plasmid. And the neomycin cassette is going to be replacing exon number 2. And therefore, we have a recombination process that took place. We replaced exon 2 by neomycin cassette. And therefore, if we add the gene, if we add the antibiotic, the cells will survive. This way, we already have embryonic stem cells that contain a knockout of that gene. Okay. Now, the third possibility is that there is a non-specific recombination, meaning in the genome, unspecifically, not only to that gene, so that it doesn't take into consideration the homologous arm of that gene. It took homologous arms from nowhere, and then it took the gene and it took the thymidine kinase with it. In this situation, the gene, the neomycin cassette, and the thymidine kinase will all go into the genome. And therefore, if we add a drug that is activated by the thymidine kinase, we can kill those cells. So that's why we have the thymidine kinase downstream, because we want to know if unspecific recombination like this one happens away from the gene, it would take this small part with it. And therefore, if this unspecific recombination took place, we could kill those non-specific recombinant cells by adding the drug for the thymidine kinase gene. Now we have 3 different cells in the culture. Those will directly die by neomycin because they have no resistance. Those, they will resist neomycin, and they are not sensitive to the drug that is activated by thymidine kinase because the thymidine kinase was not recruited, since the recombination is specific only to the green gene. And the third situation, we have the cells where we have both cassettes, and therefore these will die with the thymidine kinase gene. So the selection is very easy because these two are going to be wiped out and only the red cells will survive. Those are the ones that I will take into the next step. Okay. So all this, what I'm saying right now, is written extensively in the text here. So we said, first we collect the stem cells. First, let me start from the beginning. So we have in our vector the thymidine kinase gene and the neomycin cassette gene, and the homologous arms, the homology arms. First, we will take the embryonic stem cells. We will transfect our vector there, and then we will have 3 different products. With the right homologous recombination, it would be this situation here. And the end up is the replacement of the exon that you want with the neomycin cassette, and therefore only neomycin resistant. The non-specific recombination will carry the thymidine kinase, so it will be this condition here, and therefore if we add the drug that is activated by the thymidine kinase, it will kill those, and that's 4B. And the third one, there is no recombination, and therefore there is no neomycin resistance, and these cells will die. And I will end up only with these red cells. Okay, it seems that this slide should have been before, but it flipped here. But anyways, I was going to describe the positive and the negative selection that I mentioned already 5 times right now. The positive selection is the usage of the neomycin, and the negative selection is the usage of the thymidine kinase. Okay, so what is the next step? The next step is that we will take those cells and now introduce them into another, sorry. It seems that this slide has been duplicated, but again, I'm repeating myself and again with the same information. So the vector, the genome, and then we will have the end product, which would be the cells containing the neomycin cassette only. Okay. So the next step now. The next step is to breed a totally different types of animals, because we were using the brown, now we are using the black. We mate black males and females, and that female has an embryo now at the blastocyst level. It would be the perfect timing to add those engineering red cells where we interrupted the gene. And then we take that embryo and we implant it into a pseudopregnant female. And that female will generate chimera, where some of the genes come from the brown mouse and some of them come from the black, and that's why you have this striped animal. This would be the first generation. As I said, you have to grow this colony, so we breed this animal with another animal, and we will have offsprings. Obviously, the embryonic stem cells that come from the brown animal are the ones that we are interested in, because these are the embryonic stem cells that has knockout of the gene. And therefore, any black animal that comes in our colony, we don't use it. So we breed this to that black. If we have a black animal, we know that it's not a knockout directly because it doesn't have the embryonic stem cells from that brown animal. Then we will take the brown animals, and you can see here, this animal is heterozygote, meaning it's carrying one copy of that transgene or the knockout gene. And this guy also have one copy. If we breed them together, we will end up having an animal that has 2 copies of the neomycin cassette, and therefore here we achieve deleting the 2 copies of the exon number 2, and this is the knockout animal. This is the whole body knockout of that gene. So first step, embryonic stem cell. Second step is more or less similar to the transgenic that we discussed with the DNA injection, but it's at a different stages at the blastocyst. Why it's at the blastocyst? Because it's the best stage where we can reintroduce embryonic stem cells back. And again, we use embryonic, we would use the Southern blotting or the PCR to identify the transgenic animals. Great. After that, we need to perform the genotyping and the phenotyping. The genotyping is to look in the genome and examine if we have inserted that DNA into the genome. Phenotyping means to look what is the effects of that target gene. We deleted a gene, so what would be the effect? Some of the phenotyping results could result probably in killing the animal. So if you remove an exon, you remove the gene, during embryonic development, the animal dies. So this is lethal knockout. Some of them will have no effect or moderate effect. One of the examples I put for you here, the tumor suppressor gene p53. The p53 should block the cancer formation. So in humans with the defects in the p53 causes susceptibility for certain cancers. If we remove p53 from mice, the mice survive, so there's no lethality, but then when they grow up, they grow a lot of tumors. So in other words, I know the absence of p53 causes tumors. So that's the end up. And you can study this in terms of drug. You can find anti-cancer drugs and stuff like that here. Now, one last slide or two slides left. Let me see how much time I still have. 3-4 minutes. Okay. Can you still see my slides? Could you please let me know if you see my slides? Okay. Okay. So now this is a concrete example where we generated a knockout animal for the sarcoglycan protein. So the targeting vector, so if you look here, you have the genomic locus. We know that it has some restriction enzymes like HIND3, NOT1, EcoR1. We know the exon number 2 is very important. And then we generated a targeting vector where we use the area between the sequence between HIND3 and EcoR1 as the first homologous arm. And the second part is between just after the HIND3. This is the other homologous arm. You can see the thymidine kinase gene is located here. Now, in the targeting vector, we replaced exon number 2 with a neomycin cassette. Okay. And when we do that, we lose this EcoR1 site. So if we cut the genomic DNA, we don't get digestion anymore here. Okay. Now, after generating the animal, we will have two outcomes. Either the animal is still wild type, so we'll have this type of product, or have a copy of this and that. This would be heterozygote, or both copies of this, which is a homozygote. So first, let me explain for you why we have this EcoR1. It's very important because if we make a probe and do Southern blotting, remember to do a Southern blotting, you have to digest the DNA with and restriction enzyme. So if we use EcoR1 to digest the genome of that mouse, we will have two different sizes of the probe. One that starts from here all the way there. And this would be the knockout animal because we lost the restriction site there. It will be only this size. And the wild type, which will have two different sizes, this guy and that guy. So if we look on the Southern blot that is presented, you can see that you have both. In the wild type, you have only one band. And in the knockout, you have two bands. One corresponds for the wild type, which from here to there, and one that comes from the knockout animal. Okay, from the knock-in gene, I want to say, the neomycin cassette gene. Also, you can see that we can use PCR. So you have a primer that sits only on the neomycin cassette, and a primer number 3 that sits on the genomic DNA. And the primers 2 and 3, they are number 2 is only in the exon number 2. So in other words, if I amplify a product between 2 and 3, it will come from the genomic DNA from exon number 2. And if I have a product from 1 and 3, it will be because the neomycin cassette is there. So a wild type animal would have a product like this. It comes from 2, 3. And a knockout animal, it will have a product from 1 and 3. And a heterozygote, which have a copy of each, will present one like this and one like that. So you have two copies And that's exactly what you see in the embryonic stem cells. So this is exactly what you see in papers in terms of transgenic animals. Now, if you have questions about this particular slide, I can come back next time. If you have any general questions, I will stop right now and I can take those questions. So first of all, let me stop the recording. So make sure